Hello, in this video I will talk about a biochemical test that is used to characterize enteric bacteria on the basis of their ability to utilize citrate as the only source of their carbon. Every organism requires energy to perform their metabolic activities. So does with microbes. They also need energy to perform their metabolic activity. For that, carbon is important. And this test will find out the ability of the organism to utilize citrate as the source of their carbon for energy generation or for performing their metabolic activities. So without any further delay, let's get started. This test is used to differentiate among enteric bacteria on the basis of their ability to utilize citrate as the sole source of carbon. The enzyme citrase produced by the organism breaks citrate to oxaloacetic acid and acetic acid, and these products are later converted to pyruvic acid and carbon dioxide enzymatically. So as I have mentioned, these organisms are going to utilize citrate as the source of carbon, and this enzyme citrase is produced by these organisms to utilize citrase because this enzyme will break citrate to oxaloacetic acid and acetic acid, and this oxaloacetic acid and acetic acids are later converted to pyruvic acid and carbon dioxide enzymatically. Principle. This principle can easily be, it's very easy to understand if you see this picture. These are the test tubes. One is with, actually these test tubes are having slants. This media are, a slope, like, you find a slope here. These are called as slants. So these media are in the form of slant, and one media, which is represented as I think A 10 to the power of -4, is inoculated with microorganism, and the other one is not inoculated with any microorganism and thus taken as control. Okay, so for every biochemical test, it is important to take a control slant or control media. This test is performed by inoculating the microorganism into an organic synthetic medium. So as I have mentioned earlier, the organisms have been inoculated in this medium. And one indicator, very common, bromothymol blue, is used. So when the citric acid is metabolized, the carbon dioxide generated combines with sodium and water to form sodium carbonate, an alkaline product which changes the color of the indicator from green to blue, and that constitutes a positive test. You see, but in control green remains as green, but here it changed from green to blue, so it means the citrate utilization test for this isolate is positive. Requirements: broth culture, that is organism or isolate, Simon citrate agar plates, Bunsen burner, and inoculating needle. Prepare Simon citrate agar slants maintaining the pH 6.9. Dissolve all the constituents except phosphate, which is to be dissolved separately in 100 ml of water, and make a volume of 1 liter. Pour the medium in the culture tube and sterilize by autoclaving. Prepare the slants. Inoculate Simon citrate agar slants with isolates by stab and streak inoculation. Take a slant as control. Incubate the slants at 37 degrees centigrade for 48 hours. So as the procedures you need to follow, and by that you can conclude that whether the isolate is capable to produce citrates that uses citrate as the sole source of carbon. So these procedures you can take a note of it.